The International Boundary and Water Commission's Texas Clean Rivers program for the Rio Grande Basin monitors water quality in the Rio Grande and its U.S. tributaries. Bacteria data provides information about whether the river is safe for swimming and other kinds of contact recreation. Bacteria levels are high in the Rio Grande in most urban areas in Texas, including Laredo, where the IBWC conducted a special bacteria study in 2011.
The state of Texas uses E. coli bacteria as the indicator for recreational use, while Mexico uses fecal coliform. Because the river is shared by both nations, both E. coli and fecal coliform have to be analyzed. This video explains how the analysis is completed for the different kinds of bacteria. All samples that arrive at the laboratory have to have proper documentation, called a chain of custody, which has information about the samples such as when and where they were collected, who handled them, and who received the samples at the lab and at what time. This is important to ensure samples are analyzed within their required holding time of 8 hours.
The method Texas uses to analyze E. coli bacteria is called the ColiAlert method. Here, staff from the City of Laredo Health Department Laboratory prepare the samples of the bacteria special study in the Laredo area. A pre-prepared media powder is mixed with the 100 milliliter sample, shaken to dissolve and ensure a homogeneous sample, and poured into trays with 49 large wells and 48 small wells. The trays are sealed so that bacteria will be isolated in each well. The trays are incubated for 24 hours at 35 degrees Celsius. E. coli values are read by inserting the incubated trays under a UV light. Wells that glow are positive for bacteria colonies. The glowing wells are counted, and then total values of small and large wells are looked up on a statistically derived chart that gives the most probable number of bacteria colonies per 100 milliliters.
This sample has 1 small well and 11 large wells, which translates to a value of 13.4. The standard for E. coli in Texas is 126, so this sample is meeting the standard. This sample, however, has all wells illuminated and has a bacteria value of greater than 2,419, which is well above the standard. Both of these samples were collected in the Rio Grande in the Laredo area. Since Mexico's standard for bacteria is expressed in terms of fecal coliform, the lab also performs analysis of fecal counts, which is more complicated and time-consuming than analyzing E. coli. The samples have to be filtered through membranes and cultured in petri dishes. Here, Rebecca Castro from the City of Laredo Health Lab performs various dilutions on membrane filters.
She is following procedures required by the National Environmental Laboratory Accreditation Program to ensure quality results. Bacteria levels in some parts of the Rio Grande are so high that the only way to get a count is to dilute the sample. Each membrane has a known concentration of the diluted sample filtered through it. Any fecal coliform bacteria that existed in the filtered sample remain on the membrane, which is then placed in a petri dish with media. The petri dishes are sealed in a bag and then placed in a water bath to be incubated at 45 degrees Celsius for 24 hours. After the incubation period, lab staff set out the petri dishes and count the bacteria colonies that grew on each membrane by counting the blue colonies in each grid. Diluted membranes are multiplied by the dilution factor to get a bacteria count per 100 milliliters of water. The laboratory procedures to calculate bacteria levels in the river water can be complex, time-consuming, and expensive, but are necessary to determine whether the river is acceptable for recreational activities such as swimming and fishing. Results from the lab analysis are posted on the IBWC Clean Rivers Program website. The IBWC appreciates the support of its partnering laboratories, such as the Laredo Health Department lab, who performed the bacteria analysis for water quality monitoring in the Rio Grande.